Hi, I'm Colleen Rosales. I'm a PhD candidate and research assistant at Indiana University. I do atmospheric research and air pollution studies, and my talk is mostly how about I try to make a program and make my data analysis more organized. So these data that are coming from these collaborative field research and field campaigns are the data that I analyze using this program that I created with the Wolfram language. Hi everyone, I'm Colleen Rosales. I'm a PhD candidate and a research assistant at Indiana University. And today I'm going to talk about how I created a program for a data intensive field campaign. So my talk will be consisted of the following. So first, I'll define what a field campaign is and what are the computational needs for such a field campaign, and why I decided to choose Mathematica or the Wolfram language to fulfill these computational needs. And lastly, what are the tasks that I've completed using this program that I've created. So first, what is a field campaign? Field campaign in atmospheric research are observational studies planned for a specific location at a specific time period during which measurements can be conducted from airborne, or in my case, ground sites, to study physical and chemical processes in the atmosphere. This is usually a collaborative, multiple research group effort, and so you can imagine the amount of data that is collected in these types of collaborative research efforts. And examples of the field campaigns that I have participated in are the following. So first is Ironic 2, or which stands for Indiana Radical Reactivity and Ozone Production Intercomparison. This was done at a forest in Indiana, wherein multiple research groups intercompared the instruments that they have been using to measure this certain chemical species. The next field campaign that I have participated in is called HomeChem, or House Observations of Microbial and Environmental Chemistry. So as opposed to the previous campaign, which was an outdoor campaign at the forest environment, this campaign is an indoor environment kind of campaign. And what we did was simulate activities, everyday activities such as cleaning and cooking, and look at the emissions that have been coming out from such activities, and basically track the chemistry that happens during these events. It's a very big collaboration between about 20 research groups. This was done in Texas at kind of like a bubble house, if you will, wherein there's a lot of these instruments that have been measuring different chemical species all at the same time. The most recent one that I've been involved in is called IRACE, or the Indiana Radical and Aerosol Chemistry Experiment. And so this field campaign is kind of a combination between the outdoor and the indoor field campaign. So we're looking at how the outdoor environment is affecting what's coming inside the indoor environment. And so this was a collaboration between Purdue and Indiana, wherein we looked for the first 2 weeks of the campaign, we were mostly looking at outdoor emissions, and then we go back inside and simulate experiments and see how the outdoor emissions are affecting the indoor activities. So before I dive into the program that I created for the field campaigns that I have been involved in, I want to introduce you to our home-built instrument. This is the LIF-phage instrument. LIF-phage stands for laser-induced fluorescence assay by gas expansion. And I'm not going to bore you with the details of how it works, but basically we use this instrument to measure a certain chemical species. And since this is home built, there is no manual or software that comes with it, and we'd have to analyze the data ourselves, basically analyze the raw data that comes out of it and then transform that into something that would be more meaningful for researchers. So for the data collection in all our data field campaigns, the frequency is about 1 to 2 points per second. Different categories such as online or offline wavelengths and photon counts, pressure, voltage, wavelength, flow rates, etc. All these are collected at about 1 to 2 points per second. So you can only imagine how much data is generated, let's say in a month-long field campaign wherein we're running about 12 to 24 hours a day. So that's a whole lot of data to analyze by hand. And not just the data collection itself, but also you have to do a lot of calculations afterwards to get a meaningful number or a meaningful information out of it. And so what we're mostly concerned with is the concentrations or the amounts of the pollutants or of the chemical species that we're measuring. But what comes out of the instrument is voltage or photon counts, and how do you convert that into concentration? And so there's a lot of operations that are required. And so if you have a data of, say, about 1.5 points per second for about 12 to 24 hours a day for a whole month, that's going to be a whole lot of data processing, and that's going to take a whole lot of time. Thankfully, someone in our lab, a former researcher in our lab, devised this MS Excel /VBA program. And so that made the processing a little manageable. And so it had a GUI, and it had this fill-in forms and everything. However, as we progressed, it became more slower, and also it took a lot of time for it to analyze the data. It took about 30 minutes for the VBA program to run, and at some point you do want to make your data analysis faster. And so it was useful for a certain time where you could do quick look of data. It was helpful for saving output files as individual sheets. And so you can kind of see each sheet how it was calculating. But the drawback was that it was running so slow. So imagine manually copy-pasting data files. It's like basically what the VBA program was doing for me. And so I wanted to eliminate that process, the manual copy-pasting, and basically just give me the concentrations, like the table of concentrations and all that, and have the analysis run faster. And so that's when I looked to another program, and how can I make this data analysis faster? That's how I chose Mathematica and the Wolfram language. So I wanted to make the data processing faster, traceable, and such that not just I'm copy-pasting and I forget what I just did. And so some of the advantages that I see of the Wolfram language is that it's a natural language input. So prior to this, I didn't have a lot of knowledge in coding. And so it was very convenient for me that you can have this natural language input. And in addition to that, the documentation is really helpful. There's complete examples, and you have this debugger when your code doesn't run. And so those two were very helpful for me in trying to navigate Mathematica. Also, it has a document-based workflow. So all in one window. You have your programming, visualizing, calculations, text, and all in one window. What was also very useful for me was the set delayed functions, which allows me to define my own function and just apply that to my data set and have it run. And then my most favorite part function would be the buttons and fill-in forms, which makes it more interactive in a sense and more friendly to introduce to other users as well. So the tasks I've completed using Mathematica, the biggest one of all is creating this Extractor program that extracts the data files out of our instrument. And embedded in this program are consolidation of multiple files and matching data files from different sources with different formats. So in a field campaign, not every research group has the same data format. Some will be in CSV file with how many columns that you don't want all those columns, you just want the concentration column or something like that. And some will be in Excel format, some will be in some unreadable format. And so that was helpful for me. This program was helpful for me in trying to match these data from different collaborators. Also, what was helpful was that since, for example, us, we take our data every minute, or I'm sorry, every second, but some other groups take their data every minute or every 15 minutes. And so there's that interpolation function, which we can look at more into detail later on. But that was one of the functions that was helpful for me in terms of consolidating data from other groups with mine. So some of the things that my extractor can do, first is to filter irrelevant, blank, or not a number fields. So by using the functions position and delete cases. So there are other functions that I use as well, such as cases, but I think it's easier to use delete cases if I don't want those points anyway. And also what I think is nice about this is that you keep the raw data and you just take parts of the raw data and then process that and take the information you need from the filtered data. And then we have online and offline points wherein I have to subtract the offline from the online points. And so before I do that, I have to average all the online and the offline. But in a sense where, so it's sequential, online, offline, online, offline. So if I want to kind of separate the online from the offline, I don't want all the online to combine and then all the offline to combine. I want, at a certain time point, this online minus this offline. And so what was helpful for me in that task was the split by function. And then some more common things for averaging, like mean and standard deviation. I also have here the pick function. So wherein you kind of compare two lists, and if in list 2 something appears, then that function picks out that element. I also want sometimes to calculate values just for the online but not for the offline, in which case I use the if function, or for multiple if functions, I use the which function. And sometimes in a certain block or sheet of online or offline data, I don't want all of these data points. And so I use a defined function, a set delayed defined function that I defined, wherein I drop the first and the last points. And then this data matching and interpolation was very convenient because basically you just say or define a time series and then call the interpolation function, and then it gives you the function wherein you can input the timestamps, and then it will give you the data point for that timestamp. So that was very convenient. We also have, so once I'm done with all of these data analyses, I can easily export and consolidate these files. using the append to function. So sometimes what I'm doing is divide the data by 6-hour chunks and then extract each 6-hour chunk. And then, so with the extracted file, what I'm doing is like importing it and then using a master list. So append to that master list and then export that master list into one big CSV file. Also very useful for visualizing the data that we're getting is the date list plot. So this specifically is for like time series plots. Like say, for example, we want to see chemical concentrations in a day. And so these types of plots would be useful for analysis of air pollutants, say, for example, in a day. So this is the interface of the Extractor program. Some information at the top and some instruction for specifically importing water values from other research groups. And what I really like is that I can just have this button, like I can just click this button. So I hide here the code so that anyone who uses this extractor wouldn't be intimidated by all these code. But what's cool is that from the VBA code that I translated, so that one was like about 500 pages of code, and this doesn't seem like it's 500 pages. And so that's pretty cool. And also I can just hide that code and just show this button, which shows another window with a lot of buttons. So you see, like, I really like that functionality. And so the green ones are the kind of the more important buttons, and the gray ones provide just more information about the program itself. And so I guess we can try and run and extract a file from our instrument. So initially we can set parameters, and this is a fill-in window. We'll just input some information. So let's say laser power. So these are usual values that I would input. So there's specific formats that the code translates into a numerical value. And then I can say, pick the file. Might be in here. So let's say I want to analyze. So our file types are .out files, which aren't usual file types. And so we can try and pick this 12 p.m. out file. And so initially, it would take about 30 minutes. On my computer, it would take 30 seconds, but a while ago, I tried it here. It took 15 seconds to run it, so it's pretty cool. So you can review the parameters as you input them. You can also clear that display. And then we can extract that. And while it's extracting, we can explore more. So here is some description about the steps, specific steps that the extractor is doing, variable names, and then what I didn't have here are the definition. Oh, there we go. So it took 19 seconds. This one shows a preview of all the results. And so we have here average information. And then the concentrations that are of interest to me are here in the last 3 rows. So this is a preview, but if you want to view the full results table, this is right here. So I think this is calculating per minute. So there's that calculation. And so if we wanted to visualize these values, so these are date list plots embedded in these buttons. See if it will work. Take a little bit. Yes, there we go. So this doesn't show all of my peaks, but yeah, so these are periods where events happen. these rise in concentrations are, where events happen. And so those are the ones that we're most interested in. So it's very handy for a quick look of the extracted data. And then, so you can export those plots, and then you can also export the summary, like the results file, or you can also just choose to export the concentrations and all of them in a CSV file. And so if you exported those, there is this compiler, if you may, that will compile all those extracted files. And so say, for example, so I wanted to compile all these concentrations in one big file. I could do that. Let's just say 2018. And then that's going to consolidate all those files. Yeah, it's going to tell me to save it here. And so that was very quick without the pain of opening each Excel file, copy pasting, and all of that. And all of that with just the append to function. So that's pretty neat. And yeah, so that's how I expedited my data analysis for these big field campaigns. And I'm probably going to still use this program for the next few field campaigns. I've started telling my lab mates about this program, so that will help them expedite their own data analyses as well. And with that, I'd like to end my presentation. I'd like to thank the following. The Indiana University Provost Travel Award for Women in Science, the High Performance Systems at IU, my lab, Nicholas Bronk, who introduced me to Mathematica. And if you have any questions, I'll be open to them. Thank you.